Hello, ISMB. I am Jaime Moreno, Junior Research Scientist in Novo Nordisk and former student in the Copenhagen University. Along this project, I will explain my master thesis, Identification of ligand-receptor interactions in CKD and NASH, by integrating single-cell and spatial transcriptomics that I performed with the Professor Alvin from the University of Copenhagen and Gianluca Vivek, Senior Research Scientist in Novo Nordisk. The main goals of this project is firstly to create an analytical framework using machine learning tools to identify ligand receptor in these two complex diseases, chronic kidney disease and non-alcoholic steatohepatitis, by combining these two technologies. And secondly, investigating the progression within the disease by breaking them down in the stages and in order to gain potential therapeutic targets. A little bit of background. CKD and NASH are two complex diseases with fibrotic insult and associated with mortality. Over the past few years, significant advancements have been made in the field of single-cell and spatial transcriptomics. While single-cell allows us to study cell population, spatial transcriptomics adds the three-dimensional information, enabling to explore gene expression within specific tissues. By integrating this information, we can identify the cell types communicating and which ligand receptors are involved in this process. In addition, comparing communication between the stages of the disease, we could understand how the progression works and optimize target identification. Firstly, we carry out separate analysis for each technology. In single cell, we follow the standard pipeline of normalization, clustering, and annotation. Then, we use the statistical tool CellSat to infer all the cell-cell communication for each disease state. On the other hand, we create a workflow for the spatial data, which consists on integrating all the slides using Harmony, where we ignore the three dimensions for this part of the analysis. Then we deconvolute the output using search location in order to see the cell types present within the spots. Using a simple k-means algorithm, we perform a composition cluster analysis, grouping the spots with similar cell types proportion. And finally, we calculate the co-occurrence using the tool ISMB, which determines if the frequency of two cell types appearing together in a composition cluster is higher than expected, indicating a potential interaction between them. Now that we have gathered all the necessary components for our pipeline, it's time to combine all the information. We begin with the cell signaling data from single cell. Then, in the second step, we focus only on these ligand receptor interactions whose cell types are positively concurrent, surpassing the expected frequency. Afterwards, we assess if the ligand receptor are differentially expressed when comparing two disease states, such as healthy versus stage 1 of the disease, for instance. Finally, we took these selected ligand receptor pairs and visualize them within the spatial slides to see if they are co-expressing in the same regions or not. This slide is just to clarify steps in the workflow. The box plot shows the proportion of the cell types within a compositional cluster. In order to remove noise, not all the compositional clusters were taken, only those with high proportion of the relevant cell types depending on the disease were kept. The table is an example of the differential expression analysis. A ligand receptor pair is up or down-regulated if both genes are differentially expressed in the same direction. This ensures to only give the more promising targets. Let's take a look at some of the kidney results. This is the communication between energy3 with ERB4. This ligand receptor pair was found to be always upregulated in the disease. We observe the cell type communicating under the column source and target, and on the left the different stages, having healthy, CKD, and AKI, which stands for acute kidney injury, known for being an intermediate state between healthy and the chronic state. Interestingly, looking at the table, we see how once the chronic state is reached, the same cell types appear in both comparisons. Particularly, the proximal tubule cells are always there. This may indicate that this ligand receptor pair could be an interesting target for addressing the chronic state in proximal tubule. In addition to examining common ligand receptor pairs across all comparisons, we also focused on single differential expression analysis, such as healthy versus NASH. We discovered that the communication between PDGFC and PDGFRA is always upregulated in the disease between cholangiocytes and stellate cells and myeloids with stellate cells. When we look into the image, we see the raw tissue on the left. If we look deeper, we can identify some darker regions. This darkness is indicative of extracellular matrix accumulation resulting from fibrotic inflammation. The next two columns show the co-expression of this ligand receptor pair, and the last column corresponds with the co-expression of well-known fibrotic markers. If we compare all of them, we see a clear overlapping between them, suggesting that PDGFC and PDGFRA are involved in the progression of NASH, or at the very least the association with the development of fibrosis. Along this project, we have demonstrated the power of combining single-cell and spatial transcriptomic data. By integrating this information, we are able to harness the strengths of each technology, leading to a more comprehensive and robust analysis. This analysis not only enables to identify ligand-receptor interactions, but also the cell types in which they are communicating, providing more details of the progression of the stages in CKD and NASH. Thank you everyone for listening, and don't forget to stop by on Wednesday between 6 and 7 for further discussions.